Antibodies were among the first proteins to be recognized as therapeutic agents. Antibodies represent very attractive therapeutics that fight disease as part of our body's natural defenses. However, it has taken well over a century to optimize and tune the use of antibodies as therapeutics. Nowadays, they provide meaningful treatments for many serious diseases, helping millions of people. Genmab is a biotechnology company that works to improve the lives of patients by creating and developing innovative antibody products. It is our passion to make the best possible products and develop these for the treatment of cancer. Genmab is proud of its expertise in the antibody field and of being at the forefront of antibody-based drug development. Our product, ofatumumab, is now marketed as Azera by our partner Novartis and has been approved for treatment of certain chronic lymphocytic leukemia indications. Daratumumab is being developed by Janssen Biotech under a license agreement with Genmab. Daratumumab has been approved in the United States and Europe for the treatment of patients with heavily pre-treated or double refractory multiple myeloma. It is in clinical development for treatment of additional multiple myeloma indications and non-Hodgkin's lymphoma. We develop these products to optimally exploit the body's own defense mechanisms to fight cancer. Yet there is still room for improvement and new treatment options, as current therapies are not always sufficiently effective. This means that there is an urgent need for novel treatments that benefit more patients.
It is our philosophy that successful drug development involves a rational and knowledge-based approach. Antibodies are very good in protection against disease, and that's why we continuously study and explore the mechanisms of how antibodies actually function in immunity, and we use this knowledge to create next generation antibody technologies and also to create and develop drugs that can treat human disease in a much better way than earlier generations of medicines.
One strategy to optimize therapy is to arm the antibody with a chemotherapeutic agent. These products are called antibody-drug conjugates. The antibody delivers the toxic agent specifically to the site of disease, and the malignant cells die. Genmab's first armed antibody is currently being tested in clinical trials. For this product, Genmab has entered into a research collaboration agreement with Seattle Genetics to use its drug conjugate technology. Most diseases are caused by multiple factors and have a complex pathology. Therefore, another approach is to use antibodies to attack tumor cells in two places simultaneously, or to attract specific immune cells. This can be achieved by using bispecific antibodies. This type of antibody is at the frontier of antibody drug development. Genmab developed the DuoBody platform, which is an innovative technology for the generation of bispecific antibodies.
In our research, we like to be inspired by nature. So in the breakthrough discovery, we found how human antibodies can form bispecific antibodies in vivo, and then use this knowledge at the molecular level to generate the DuoBody technology. So I'm very proud of the highly talented group of scientists that helped us to then transform this basic knowledge into the DuoBody platform.
The DuoBody technology allows us to make bispecifics from any pair of antibodies. The two antibodies, each containing only a single mutation, are produced separately. After mixing, we bring these together under well-controlled laboratory conditions to create bispecific antibodies. We developed a robust, high-yield process that can be directly applied to all scales of production, from early drug discovery to development. The DuoBody technology opens up many new possibilities for therapy. Bispecifics that target multiple pathways can potentially combat tumor cells more effectively and prevent resistance to therapy. Alternatively, we may generate bispecific antibodies that direct immune cells in the blood to specifically kill tumor cells. A third strategy builds on our long-held interest in how complement interacts with antibodies.
The complement system represents a very important mechanism of defense of our immune system. And many researchers around the world have been searching to understand this mechanism and to harness its full potential for therapeutic use, for instance, to destroy cancer cells.
We investigated how antibodies activate complement at the molecular level. We built on this knowledge to develop a new technology called the HexaBody platform. This is used to create potentiated therapeutic antibodies. The HexaBody technology enhances the formation of antibody hexamers after target binding at the cell surface. Thereby, it can maximize the killing activity of antibodies. This is achieved directly or through the activation of complement. To stay at the forefront of biotechnology, Genmab collaborates with some of the best research groups in the world. For example, we work together with the Scripps Research Institute and the University of Virginia Medical School in the United States, as well as Leiden University Medical Center and the University Utrecht in the Netherlands. Together, we studied the molecular interactions between antibodies and complement. We combined state-of-the-art techniques to understand how antibody structure relates to its function. These technologies have enabled us to step up our efforts of developing antibody products for the future. For all our products, we have set up high-tech and streamlined approaches for the generation, production, and characterization of antibody candidates. The final candidate is produced using well-established methods in the amounts and quality required for clinical development. Our expert antibody knowledge enables us to develop new therapeutics. With these, we aim to improve the lives of cancer patients. Our team of experts work with passion and drive to create and develop innovative antibody-based products for cancer treatment and turn science into medicine.